Raegan Adams from GDD and Dr. Rachel Bailey's lab is next. And she is presenting Opening the Door to SLC 13A5 research.
Imagine a newborn baby, perfectly healthy one moment and then seizing within hours of birth. This is a reality of parents with children affected by SLC 13A5 citrate transport disorder, a rare developmental disorder where patients have severe seizures, intellectual disability, mobility issues, and require 24/7 care. SLC 13A5 makes a transporter protein. Think of this like a molecular doorway that brings citrate into our brain cells. Citrate isn't just part of your everyday orange juice, but it's actually a vital ingredient in things like energy, metabolism, lipid synthesis, and even neurotransmitter balance. When SLC 13A5 doesn't work because of mutations that actually break this transporter, citrate can't properly enter our cells. The result, we have too much citrate outside our cells and not enough citrate inside. Using a mouse model that develops the exact same disease as patients, I seek to understand how this citrate imbalance actually causes seizures. See, our hypothesis is that citrate could be disrupting how our brain cells regulate activity. Our brain does a pretty good job of keeping a careful balance between signals that excite and signals that calm activity. And we think that citrate might be binding to key regulators of this, causing a disruption in signal, which could cause seizures. In SLC 13A5 mice, I've been able to show that like patients, they have increased seizures and increased citrate floating around their brain. I've even taken this a step further and actually given these mice a diet of even extra citrate, and this has caused even further increases of citrate in their body and even further increases of seizures, which could suggest that citrate could be one of the culprits of this disorder, though further studies are needed. The goal, however, is not only to understand more about SLC 13A5, but to develop therapies that actually impact patients. Most patients take between 2 to 3 anti-seizure medications with varying levels of control, creating a great need for better therapeutic options. In the lab, we use gene replacement as a therapeutic strategy. In this, we use a special harmless virus that actually encodes the instructions to make these functional citrate doors, and we can deliver these to the vulnerable cells. In mice, this has been shown to decrease citrate, be safe, and to improve seizures. And excitingly, this is actually entering clinical trials at UT Southwestern starting next year. So all in all, we hope that this research and this therapy will have transformative effects for patients affected by SLC 13A5. And more broadly, we hope that this research will open more doors for collaborative rare disease research everywhere. Thank you.